We are fortunate to have Dr. Christopher Breder provide our next lecture. Chris currently serves as a reviewer in the Division of Neurology Products at the FDA. His interests include rare neurological diseases. He also teaches at Johns Hopkins and the NIH. Chris received his MD and his PhD in neurological science from the University of Chicago and completed his residency and clinical fellowship at Johns Hopkins. He did postdoctoral studies in molecular neurobiology, where he also completed at the University of Chicago and Johns Hopkins. I'm confident you're going to enjoy today's lecture.
Hi, my name's Chris Breder. I'm a medical officer at the FDA in the Division of Neurology Products and also a faculty in the program for Advanced Academic Programs Regulatory Science at Johns Hopkins University. And today I'll talk to you about a very special topic, which is the clinical analysis of adverse events. And adverse events are very important because clinical research as a whole is a balance between risk and benefit. And the currency of risk is the adverse event. It's actually how you monitor for the risk in clinical research. So I hope you enjoy today's lecture. The views expressed in the talk today represent my opinions and do not necessarily represent the views of the FDA. So today we'll use a question-based approach. It's always good when you're hearing a lecture to fully understand what it is you're hearing about. So first we'll define adverse events. And then I found it's also useful to understand why we collect adverse events. That really tells the story for the rest of understanding why and how we monitor them. And then we'll go on to how, which many of you want to know, and when they need to be monitored. Before we get into the substance of the lecture, we'll give you some definitions that are very important. And the first, of course, is the definition of adverse events. And as you can see from the notes in parentheses, this comes from the federal regulations. An adverse event means any untoward medical occurrence associated with the use of a drug, whether or not it's considered drug-related. Then the other definitions you need to know, which I will allow you to read, I won't read them through the slide, is a suspected adverse reaction. And another important concept is that of reasonable possibility. And then the final concept you should be familiar with is that of an unexpected event. To understand adverse events, there are several dimensions that need to be described by typically the principal investigator or clinical coordinator in a research study. And I've listed them here. The ones I'll talk about are designated with the red star. The first event, or the first aspect of adverse events, which is critical to understand, is time to onset. And I would say for each of these that I've noted both as a medical officer at the FDA, but also when I was in industry, that these particular issues, while they seem very simple, often cause a lot of confusion and anxiety with investigators exactly how to define them. So I hope that this particular section is useful. So there are some aspects of adverse events which seem rather cut and dry. With respect to time to onset of event, it seems very clear on the top scenario that if you have an event of one intensity or an event of just an increasing intensity, which then ends, that is one event. On the other end, it seems fairly clear that if you have an event with fluctuating intensity over a single period, if it completely goes away, that's likely or could be more than one event if it truly goes to baselines. But if that event, as one sees in the middle scenario, changes in intensity over a continuous period, that could be very confusing as to whether you should call those separate events. One example you might think about is headache, where it starts out becomes intense, almost goes away, but then comes back again. And it's not always clear whether that's one or more event. As with most of the attributes I'll talk to you about, there are very clear-cut scenarios, and there are some that are not so clear-cut, even in the eyes of the reviewers at the agency. But I would say that if you clearly define how you are designating events, and you also discuss those that seem somewhat difficult to define, that would probably be the best scenario. Another attribute which can sometimes be confusing is that of resolution, which relates in a way to the last attribute we talked about. So it's very clear that if the event goes back to the baseline state, it's usually considered resolved. Somewhat more confusing could be an event that does not change to the last inquiry. An example of that could be, well, also for the middle state, something like anemia, where it may hit a nadir but then stay the same without clinical consequence through the rest of the trial. If an event continues to worsen clinically, then that event would need to be followed definitely, and even outside the time period of the study. But for example, many adverse events simply become non-symptomatic, and one could say that if they're clinically stable, that is resolved. Severity is the next aspect or attribute of adverse events that's critical to understand, and at least at the FDA, there's a common practice to describe them according to one of two guidances. Typically, people use terms like mild, moderate, or severe, so this is not a very challenging thing, but there are other descriptors such as something known as the CTC-AE, Common Terminology Criteria for Adverse Events, that are typically used for chronically ill patients, and I've given you the reference here. For more healthy subjects who are in trials, the guidance on the describing adverse events that's in the vaccine guidance is typically used, and I've included that URL as well. The next attribute is that of causality. How likely was it that the drug caused the adverse event? And for that, people typically use words like not related all the way through it's certainly related. And then definitions are given to those different terms, so that even though they're categorical terms and they should be intuitive, it's good to know exactly what is meant by that. And I've provided you with two documents that discuss how to assign causality, and their URLs here. The next attribute that's critical to record for adverse events is known as serious adverse events. And this is actually a term that is defined in a regulatory sense. There's no ambiguity about what is meant when you check off that something is a serious adverse event. And it would include any of the following things that I've listed in the bullets here. For example, deaths, life-threatening events, hospitalization, either the initial one or a prolonged hospitalization, such as if someone gets a hospital-acquired infection, disability, congenital anomalies, or an event that requires intervention to prevent permanent impairment or damage. And then the final attribute to discuss is action taken, which is very critical because it helps you understand how serious or how severe the event was. Typically, if some side effect happens to you and nothing needs to be done except maybe resting a little, that is of less consequence than needing some other medication or some non-drug therapy like physical therapy. You could have the test drug completely withdrawn, which would be considered a very important event in a clinical trial, or the physician running the trial could actually decide to discontinue you from the trial. So it's very important to know what consequence the event had. Okay, now that we've talked about what adverse events are, I'd like to give you just a few slides about how to think about adverse events before we talk about the details of analyzing the incidence and severity of adverse events. So you'll have some context. The first consideration is that adverse events are just one of several means to assess safety. None of these are done in isolation. And just expressing my own opinion, I believe adverse events are one of the most critical or important of the methods of safety assessments, because it's what is important to the patient that gets discussed, or the complaint of the patient. The other one that I consider very critical, almost irrespective of all the others, is disposition. Disposition is whether the subject completed the trial, withdrew from the trial, and if they withdrew, why they withdrew from the trial, because it's really, as a former director at the FDA used to say, where the rubber meets the road. Once you stop taking a medication and drop out of a trial and stop getting the health care that comes with a trial, you're making a statement about that therapy. So, but the others are also very important and need to be interpreted in the context of their importance to the subject. For example, EKG changes, lab changes, or changes in vital signs, all important. And with all of these, one needs to consider the condition of the patient. For example, a change in creatinine for one patient, the same change could have a totally different meaning for another patient. The other consideration about drugs in general, pharmaceutical therapies, is that all of them bring with them adverse effects. There's a very famous person, Paracelsus, who was the father of, or is said to be the father of modern toxicology. And he said, he said it in German, but he said, the dose makes the poison. And I've given you a few examples where that's true. In the top right, you can see warfarin. In one dose is a rat poison. In another dose, up till at least very recently, was one of the few means of anticoagulating patients long-term. But also simple things like caffeine and even toothpaste have their toxicity threshold. So one should not be surprised with any drug that they have a toxic profile or adverse events, because all drugs, for the most part, do. I would also say another thing to consider here is that one can't study safety without considering efficacy. There's been some mention, especially recently, that one could do away with the measure of efficacy in the assessment of drugs, but just because of what Paracelsus has said, in the dose is the poison, you need to know what dose is efficacious to know what safety is relevant. One can always pick a dose that's irrelevant, that looks very safe, but has no efficacy. So the two really go hand in hand. And to discuss the analysis of safety, one typically starts out with a discussion of what efficacy is. It always comes first. A drug that is not efficacious cannot be analyzed for safety because it's irrelevant. And then the last general concept is that of risk-benefit, which I've termed here the adverse exchange rate. The same adverse event could have very different meanings for patients. So if I have a drug that happens to cause thrombocytopenia with bleeding events, my consideration of that drug will be very different if it's something effective as a novel cancer chemotherapeutic agent. In the same vein, it will actually not be too hard to know how to think about it if this is a drug which is the third or fourth line therapy for a chronic disease and there are other safer therapies. I'll be much less likely to want to use that drug. Okay, where it becomes difficult is, as one sees in the arrow in the middle, in the gray zone in the middle, it's harder to know how to think about that for a first-in-class drug for a serious disease, but not quite the same as a disease with fatalities such as cancer, and also the first in line for most chronic diseases. However, on the ends, it's very clear if it offers a risk. Most people with very serious and fatal diseases are willing to take therapies that have significant adverse events tied to them. On the other hand, people who are taking the third or fourth line for a chronic disease are typically not willing to take a medication that has very serious side effects attached to them. Now we'll move to the how to capture adverse events. And this is a very important topic that's of discussion, I know, around at least universities at this time. The typical way adverse events are captured in clinical trials are the spontaneously reported events. And the upside of doing it that way is that they're typically more clinically relevant if the patient is willing to say something about that. There really are unlimited domains. If something is bothering you, you can generally figure out a way to describe it. You're not limited in any means, only so much as the patient's able to articulate. You can inquire about them as frequently as possible. This sometimes becomes an issue because in some clinical trials where the visits are spaced very far apart, there could be a tendency to forget adverse effects one has. Of course, one could say then that they may not be as clinically relevant, but it is something to consider. Okay. On the downside of the spontaneous reporting of adverse events, there are and I'll start from the bottom of my list here, there are very definitely cultural differences in reporting adverse events for the same drug, for the same event, especially in domains like pain. I've noticed that to be the case, and that's very well known in the literature. It's also thought to be a less sensitive means to collect adverse events. Okay, it's only what the subject considers very serious, but for various reasons they may not complain about something which is actually very important. On the other side, we'll go over to the right side now, there are objective-based ways to collect adverse events, and they have advantages and disadvantages as well. And objective means are, for example, lab values, and those are good because you can define what you measure, and both a positive and a negative side is that they have a threshold attached to them, so you can make it very clear as to at what point an adverse event is happening. That being said, these thresholds change with a number of different issues. They change with age, so a pediatric study, for example, of 1 or 2 years duration, the subject can go through many changes in the reference range. Your consideration can change with the person's medical history and at what stage the disease is. Another not positive aspect about objective-based measuring is that you can only measure what you define. So if you measure something very specific, you could miss changes even within the same organ system within the body. Okay, and then clinical relevance is somewhere in the middle. If you happen to be measuring the exact correct thing, it could be very clinically relevant. But all lab values are surrogate assessments. They are not actually looking at the clinical benefit or clinical disability. It's an approximation of that. So the real relevance always has to be established. And then finally, in the middle, we have scale-based measures of adverse events. And an example of that in the field of antipsychotic therapy, it's typical to use a scale called the AIMS scale, Abnormal Involuntary Movement Scale. And these have some value because you can focus on some very difficult concepts, especially some patient populations have a hard time articulating certain types of adverse effects, so it allows you to probe them more. Although on the downside, again, like objective events, they can be very limited in scope, and if that's all you're looking at, you'll miss a lot of information about the subject. There can be bias in the instruction, both to the patient and to the person who is collecting the events. Sometimes that is the principal investigator, sometimes that is a caregiver, and sometimes you can direct them almost to collect any outcome you want, depending on how biased the instructions are. So it's not simple enough to know that a scale has been used. You would also want to know how the instructions were worded and also how the training was given to solicit that scale. In the middle is something that we'll talk about in future slides as well, and that are the best scale properties, or how you administer it. Okay, you should use a patient-reported outcome, a caregiver outcome, or a physician or a clinician-based outcome. In some fields, this has been studied. At least in the pain field, it's felt that patient-reported outcomes are better than the physician-collected outcomes. And then, that being said, how should you inquire to the patient? Should you give them a categorical scale or a visual analog or VAS scale to fill out? In this slide, we see an example of a VAS scale, a hypothetical example for nausea. You could go into a trial and at baseline be asked to place an X that describes the magnitude of how you feel at the moment. And this hypothetical patient has placed one right to the left, placed their X of the midpoint. And then at endpoint, or let's say at the end of 12 weeks, they've checked off how they felt, and it was just to the right of X. So this presents the problem of how to interpret collecting adverse events such as this. What's clinically meaningful if on a 100 millimeter scale, that's a difference of 10 millimeters? Is that meaningful, and does it matter where that 10 millimeters is on the whole scale? So the relevance of collecting adverse events by these methods that seem more objective can be very problematic. This is something of a busy slide. It comes from a publication that I've listed below, but it presents changes one can see with the method of acquisition. One column being either by patient interview or the next by looking through their medical records. And what it shows is that for different demographics of patients, or depending on what you're trying to collect, there can be a very big difference between what you get out of a patient interview or by going through their medical records. And so one needs to stop and think, which method will give me the most accurate picture of the patient? Am I concerned about the patient's recall and our inability to articulate? Or could it be that the caregiver or physician has possibly overinterpreted the relevance to the to the patient. So this is a real-life example of this issue. Okay. Now I'll talk about data quality. And I'll start out by saying I've been giving lectures like this on analysis of safety data and data that goes into NDAs for the last 8 years. I would say in the last 2 or 3 years, I've really switched my focus to emphasize data quality much more, because it's often more the case that the data quality has an issue on the outcome than people had appreciated beforehand. And as the proverbial idiom goes, garbage in, garbage out. You can't interpret quality into very bad quality data. So it's very important that the data that goes into these analyses is of the highest quality. So in this slide, I'd like to first discuss some technical aspects of adverse event submission and study, and that is the use of MEDRA. MEDRA stands for the Medical Dictionary for Regulatory Activity, and it is used to code adverse events for submissions to the US FDA. Now, there are different and other languages for coding adverse events for other purposes, but we won't talk about them today. As of today, if you wanted to make a submission either to the biologics or the drug evaluation branches of the FDA, you would be required to use MEDRA in your NDAs, ANDAs, which are for generic drugs, and for certain commercial biological license applications, or BLAs. Come December, they will also be required for commercial INDs, or investigational new drug applications, which is the application to study drugs in humans in the United States. Post-marketing submissions have had this requirement for somewhat longer. One may ask, although I think it's intuitive, why does one need to focus so much on data quality? And this has been studied fairly systematically, and it's been found in the study that I'm reporting here that even small deviations can lead to very significant differences in the interpretation of clinical information. The picture I have to the right provides one example of an adverse event term, and that term is procedural hypotension. And as I mentioned, MEDRA is a terminology system used to describe adverse events. It has several levels of hierarchy. It goes from the bottom, what the patient reported, or the verbatim term, up through several levels to the top, which is known as the system organ class, such as the cardiovascular system or respiratory system. And as it turns out, procedural hypotension, depending on which track you choose as the coder for the clinical study, can go into one of either two system organ classes. And so one can divert these adverse events and split them up, so the signal could actually look smaller if the reviewer is simply looking at the level of the system organ class. So this shows you just how important this is. And we'll come back to this example in future slides. There's evidence in the literature that the variability, even for very simple things, is about 12.5%. And I've noted that percent as well in my own reviews of lists of coded adverse events, the variance I had with the original versions. But it can vary actually quite high depending on the difficulty of terms. If it's a very difficult concept, then the variability can be quite high. The next two bullets discuss some of the common problems of adverse event data sets. With any data set, missing terms can present a problem, and especially for adverse events, using non-standard variable or column names can present an issue as well. For those of you who are familiar with adverse event analysis, I'm sure you've heard of the concept of lumping and splitting, and we'll talk about that in the slide to follow. So on the topic of data quality, let's talk a second about how adverse event terms get coded or put into the language you see. Typically what happens is the patient makes a statement. For example, I have a headache. That word headache is the verbatim term. Then the site coordinator or investigator will decide which term on the lowest level of MEDRA that corresponds to. And that lowest level term is called just that, LLT, or lower level term. Once the lower level term is picked, then the hierarchy for all the terms that follow are fairly set, although some terms can go into either one of two different organ systems, for example, and we'll talk about that. But pretty much the terms that follow, which is the next level, the preferred term, which is where most of the adverse events are analyzed at, or the higher level term, and then next higher level group term, and then finally the system organ class are fairly defined. So once you've chosen the lower level term, the course for that adverse event term is fairly set. So there are some jargon terms that people in the coding industry have for different inappropriate ways to code terms. And I'll start on this slide in the upper right. One is known as inappropriate lumping, where you take several preferred terms, the patient has a certain diagnosis, and you split it up into, well, the patient has a certain diagnosis that consists of adverse event terms, some of which seem very bad, some are not so bad for that particular diagnosis. And they lump them together so that you don't know that the patient has some of the very bad terms. On the other end of the spectrum, a patient could have a very bad diagnosis, and the coder could split that into many symptom terms so that you don't appreciate that they have the full diagnostic syndrome. And then the last event that I alluded to in a previous slide was that some terms can go to more than one system organ class. So if, for example, a subject has hypotension, it's very clear that that goes to the system organ class for vascular disorders. But if it's in the context of an event, they could be coded as having procedural hypotension, which could go to either the vascular disorders or the one listed in the orange ball there. So one needs to be very accurate and consistent in how one codes. The slides that follow are just a few examples of tools that are available publicly and are also used at the agency and used to look at the data quality. One tool is something known as J-Review, and if one looks in the bottom of the slide here, you can see that you can make what's known as a crosstab, which is a fancy word for a table, and that you can create rows for the different MEDRA levels. If you see at the bottom, going from the AE term or verbatim term up through the system organ class, and then you can look at that by treatment, which is in the columns, or any variable you want. And one does this because one wants to create what's known as an AE map or an AE coding map that allows you to look at how all the terms are coded. And there are at least two different ways that this is done these days. One is through the example I'll show you in J-Review, and the other uses a program that's called JMP, JMP, although you can do it in almost any type of program. So in J-Review, if you do what I've demonstrated here, that will produce an AE map that looks like this, where on the right side you have all the verbatim terms that were given in the trial, and in successive columns going to the far left side, you will see how they are assigned to the different levels of preferred term, lower level term, preferred term, higher level group term, and then system organ class. So if, as you go across, the next box is missing, that means you go up to see what group that belongs to. And this is actually a very fast way to take huge data sets and check the coding to make sure that the coding makes sense to you. Not mentioned in a previous slide, but about 15 to 20% of verbatim terms are not found in MedDRA in any given trial. And so many of these are what one calls hand-coded, where a lower level term is assigned, but it's more or less the judgment of the person coding the events. So the purpose of the reviewer is really to make sure that especially those terms that are hand-coded are accurately placed into the MedDRA system. Here is an example of the other program I was talking about. If you look on the left, you can see what could be a very complex and confusing part of a adverse event data set in the program known as JUMP, which is a SAS program. And these are not arranged in any particular order, although you may recognize from the column headers the same variable terms, AE term, which is the verbatim term from the patient, the lower level term, then the variable name for preferred term is AE decod or decode. That's the next. If you sort those terms, as you see in the lower right box, that's a JUMP sort box, by MedDRA level, you can put them in such an order that it's much easier to see which preferred terms the AE terms correspond to. And that's how one checks out the data quality in JUMP. And then finally, I had mentioned in a previous slide or shown you a picture of the MedDRA browser. The previous slide had the example of procedural hypotension. And this is an example of an investigation into the term of foggy. For example, sometimes when you take a medication that makes you feel confused, you as a patient may say you feel foggy. But if you put foggy into the MedDRA browser, which will tell you what the hierarchy will be, you'll see that the system organ class turns out to be general disorders and not anything in the nervous system. So you may choose then to recode that term into something that will group with other words like confused or disoriented so that you don't split that signal in the system organ classes. In this segment of the talk, I will discuss how adverse events are typically analyzed in pre-market submissions to the US FDA. Pre-market submissions is a long expression for what people know as NDAs or new drug applications, or BLAs, biological license applications. And then later in the talk, I will discuss the post-marketing analysis of events, or what happens after a drug is on the market. One point I'd like to make is that adverse events can come from many places in a drug development program, and I've listed them from what's typically the beginning unto the end. And I've also shown you what I believe the advantages and disadvantages at each point are. Now, because they come from different points in the development program doesn't mean they're any less important. It's just that if one is only analyzing, for example, a phase 1 trial, you'll get different sorts of information than you will from a large, simple trial. When a drug development program begins, the first study is typically known as the single ascending dose or first in human trial. And as with all the monitoring in that trial, there is very intense detail and focus on the patient. These are often conducted in clinical research labs or dedicated hospital units. The difficulty of using adverse events from these trials is that, first of all, the sample size tends to be very small, and also they're typically in normal subjects who don't have the disease. So the actual relevance to real life is not so clear, although it gives one an idea of what could happen with the drug. The second source of information are either phase 2 or 3 adequate and well-controlled trials. That's what the AWC means. And the concept of adequate and well-controlled trials is described in the federal regulations, the exact place, 21 CFR 314.126. It describes the different ways to do an adequate and well-controlled trial. The good thing about these trials for adverse events is that you're studying patients. And also, I mentioned earlier, you can't study safety without knowing the efficacy of the drug. Typically, the primary objective of these studies is to study the efficacy, so the context is just right. And because of the concept of randomization and blinding, you can decrease the bias in these trials. The downside of gaining information from these studies is that they are powered for the efficacy endpoint, which is typically much less than you would want for studying the safety. Although the analysis of safety data, the statistical techniques used do not speak to causation, rather they speak to association, the exact statistical testing. And then finally, patients may drop out in large numbers from these trials, sometimes for reasons related to the adverse effects, so you could lose a lot of valuable information from these trials. Typically, following a phase 2 or 3 trial, there's something known as an open label extension, where the trial is not blinded and it's conducted in a much more naturalistic fashion, which is good because events that happen there may be very clinically relevant, and also it allows one to see events that take some time to develop. However, one needs to realize that these are actually enriched populations because the patients who couldn't tolerate the drug have already dropped out. Also, it's unblinded, so there could be bias from both the patient and the investigator, and no comparator for context for the incidence of the event. One of the best methods of assessing adverse effects is with something that's not spoken of very much, known as the large simple trial. And it's just that. It's a large simple trial that has very few objectives and very few visits. So it can be conducted in a much more naturalistic fashion. It's powered typically for the safety event of interest. But unlike the open label trial, it's controlled. So it does not have quite the expense of the phase 2-3 trial, but one does not have the context of efficacy within that trial. So these are best for drugs where the efficacy has been established. And then finally, one can gain information about adverse effects from meta-analyses. And these have the benefit of having very large populations, and one can do many types of techniques to analyze the data. However, there are problems when one study may have an unduly large effect on the outcome of the meta-analysis. And also, pooling methodology can be very challenging. One can describe the analysis of pre-market adverse event or measure data as going through 3 phases. When one first gets adverse event data, one wants to screen the data to get the big picture. And one uses tools such as something known as the Standardized Measure Query, or SMQ, or one can perform what are known as forest plot analyses. And I'll show you examples of both of those in slides to follow. One also does what I call verifying, and that is one looks at the table of common adverse events that's submitted for the package insert or labeling. It's one of the most important parts of the pre-market submission. And then finally, one can do analyses related to hypotheses that one comes up with based on your evaluation of the data. I'll first talk about standardized measure queries, or SMQs. By definition, these are medical term lists related to specific diagnoses or medical concepts, such as hepatic failure, anaphylactic reaction, or thrombocytopenia. And I like to speak about SMQs metaphorically like a fishing net. So these are lists of about 50 to 200 terms long, used to capture events from adverse event data sets that can be many hundreds of thousands of terms long. And just like any fishing net, you may catch what you would like in the net, but you will certainly also catch other things just because you matched on one term. An example of that is the SMQ of acute pancreatitis has one of its terms being nausea. So if you cast out the acute pancreatitis SMQ and there's nausea in the data set, it will turn up as positive. However, everybody who has nausea does not have acute pancreatitis. So that is something that one needs to be cautious about. I'll also talk about something known as MAID, which you may start to see more in the literature. That's a tool known as the MedDRA Adverse Event Diagnostic System. It's primarily a tool developed at the FDA, although there's discussion of its programming in the external literature. And this is a server-based program that can take all of the SMQs, there's about 200, and analyze them simultaneously in each data set. And then the final aspect of the slide I'll point out is that recently my group at Hopkins has published a very comprehensive review on the use of SMQs for all of the approved drugs at the FDA, and I'll present a few slides on those findings in the next few slides. First, I want to just emphasize the practicality and importance of using SMQs in adverse event analysis. And for this slide, I'd like to thank Dr. Mary Doy, who was formerly a safety reviewer at the FDA and is now the lead medical officer in our Office of Computational Sciences. And for this drug, what had happened is the applicant had submitted a package insert with certain warnings and precautions. She had used the MAID server to evaluate their database and had found that the SMQ of hostility and aggression had come up with a very high relative risk. And so the final package insert ended up having a boxed warning for serious psychiatric and behavioral reactions. So one can see that this isn't just a theoretical application, but has real world use. In the publication that I described to you, all of the submissions to the FDA were evaluated for their use of SMQs for a number of terms that you can see in the table here. And it turned out that SMQs have been used since 2006, and over 184 of the NDAs and BLAs have used SMQs for analysis for a total of over 1,350 investigations. So each investigation would be for a single SMQ within an NDA or BLA. This slide demonstrates two findings from that study. The first is who initiates the analysis, and the FDA initiations are the black bars, and the industry or corporate investigations that were initiated are in the lighter bars Almost all of these are known, very few were unknown who initiated it based on the review language. But you can see a trend, you'll see in slides to follow, that the use of SMQs is increasing very dramatically. And at least at the FDA, you can see it really picked up around 2012, which is when MAID really came into use at the agency and made it, forgive the pun, much easier to do SMQ analyses. Now, one issue that's been very important to me is whether the so-called diagnosis of the SMQ was verified by either the applicant or the FDA reviewer. And by that I mean, if one gets an SMQ hit at all, it's said that that case is positive for the SMQ. So in the example of acute pancreatitis, if one simply had nausea, one could say that they're SMQ positive for acute pancreatitis, but that wouldn't mean that that person necessarily had acute pancreatitis. So we looked at whether the finding was evaluated further by looking at labs or other adverse events. And one can see by the table here that the rate of following up to verify whether something was truly positive or not is fairly high, around, I would say on average, 75%. But there are still about 25% of cases where there really was no further evaluation of the cases, so that would not be acceptable. Another item of interest was what effect these SMQ analyses had on the regulatory action during the review. And by regulatory action, I'm speaking of, did the SMQ analysis have an effect on the labeling, which is very important, or did the SMQ analysis, was it somehow related to assigning a post-marketing study requirement? And so this example, like almost all of the other trends, show that with time, there's an increasing trend towards SMQs being an important tool in the regulatory toolbox of the reviewer. Here are the conclusions from the study of Dr. Chang et al., published this year, is that, first of all, SMQs are a tool for screening rather than one used to make actual diagnoses. The second finding is that it's best to use descriptive rather than inferential statistics to describe the findings, because the adverse event data sets and the trials that they come from were not designed for this type of hypothesis safety testing. Also, more regularity is needed for people who modify these SMQ lists, and here I'm quoting language that says about the same thing for the MEDRA organization's instructions on how to use SMQs. And then finally, the last important point is that it's very important to verify the cases when comparisons are drawn between treatment groups or when one plans to use these findings for important regulatory actions. The second example of using adverse events in safety analysis I'll demonstrate is the production of what are known as forest plots. And the tool one does that is called JReview. It's been out for quite some time, even publicly. Until recently, it's been known as iReview. JReview provides many types of statistical analyses using the MEDRA language. Now there are 27 system organ classes that it does that in. And you can do this function known as risk assessment, which produces what I've termed as forest plots, which you can see in the figures on the right. And in the center, well, the first thing one notes is the column to the left, before you click on anything, has a series of plus signs, and then on the top, all of the 27 system organ classes. And then if you look on the right, you can see the the point estimate of whatever statistical variable you're interested in, odds ratio, risk difference, etc., and also the confidence intervals. The larger that circle is, you can see a rather large one about 2 or 3 lines down, the larger the population was for that assessment or in that of that system organ class. You can then click on the plus signs on the left and open it up from system organ class all the way down to, most people look at things in the preferred term level, but you can look at the point estimates and the confidence intervals for any of the system organ classes. One typically goes after those that have the largest center point estimate bubble there. And this is a very easy way to screen for safety signals in the data when one first gets a data set. Now we'll move to the adverse event tables and verifying their incidence. And you need to look at these, both the FDA does, but if you're interested in a drug and are looking at the package insert, you need to be interested in this table and try to verifying it because it's the main source of information or one of the main sources in the package insert. And often there are discrepancies one may have with the coding, but my own philosophy is that if you are going to suggest any changes, that it's really preliminary on your end. You're not with the patient, so one needs to communicate with the sponsor and agree on the most reasonable coding for adverse events. This slide shows an example in JUMP how it's done. You'll recall in the upper left before I'd shown you an example of an adverse event table with columns representing the different measure levels. One then summarizes them in a 2-step procedure to get the incidence, which eliminates double counting the same adverse event for the same subject. It's a 2-step procedure, and one can then easily, using JUMP, calculate the percent on each treatment and see which adverse events occur at a frequency greater than placebo, and then decide also on the threshold for reporting. Many adverse event tables one sees report at an incidence of 2%, but they go as high as 10 or as low as 1. That having been said, I tend to look at the entire data set because even an event that occurs at a very small frequency, such as Stevens-Johnson syndrome, don't typically happen in nature. And so even if it's a small amount, it could be very important. So you need to follow up not only on the common adverse events, but on the very important adverse events as well, irrespective of their incidence. This slide shows you a table generated in the tool JReview, also an adverse event table that's sorted alphabetically, just to show you you can generate these tables to look in almost any way you want. And this is useful if you have many events and you're interested in certain ones so that you can very quickly find the event in the table. Or you could use the same tool to present it by percent. Percent often is a better indicator of the clinical relevance, and you can decide what the cutoff is in your table and then put all of those that occur at a certain percentage and greater than placebo into your package insert. And then finally, for the pre-market analysis, here's an example of the hypothesis-driven analyses. One does these when one has small data sets. You may choose to analyze them not at the preferred term level, but at the higher level or higher level group term level. And you can also put different adverse event terms together to come up with syndrome complexes, such as is seen here. I looked at a data set for both people who had falls and people who had a higher level group term of mental impairment for a drug that caused confusion in people to see if they actually had clinical sequelae from that. Now I'd like to briefly talk about post-marketing safety analysis. And for this, I would like to thank Dr. Christian Cowell, who's a safety team leader in the group known as the OSE at the FDA, who donated most of these slides on post-market safety monitoring. We'll begin with by saying there are two main systems used now for post-marketing safety analysis. The largest system is a passive surveillance system known as FAERS, the FDA Adverse Event Reporting System, and we'll talk a little bit more about that in slides to follow. It receives very large numbers of adverse events, and since they come not from studies but from the marketed drug, it's more naturalistic. The problem being that they are very underreported. One says that at the best one gets about 20% of events that probably occur. You don't really know the denominator for most of these events because it's hard to know everyone who's using the drug, even if you have prescriber information. And the quality of the reports is quite variable. Another system which has arisen quite recently is known as the Sentinel system. I'm sure many of you have heard of that. It's also a naturalistic system with large numbers comprised of a number of centers around the country that aggregate their safety information. But as with any naturalistic system, it's hard to know all of the confounding factors or missing information that you might be receiving. As I mentioned, FAERS is the main adverse event monitoring system for post-marketing. Now it is comprised of reports that are submitted by the drug sponsors or companies. As you can see from the graph here, as with most things related to the pharmaceutical industry and safety monitoring, the numbers are increasing steadily since the 1970s. One limitation I described before is that the reporting from the standpoint of the patient is voluntary, but once the manufacturer gets the report, it's required to be submitted to the FDA, and there are specific regulations that relate to the timing of when that needs to be received based on the severity of the adverse event. As I note on the bottom of the slide, one often has very incomplete information, so it's hard to tell whether the adverse event was actually due to the product. As with the reporting for the marketing submissions, there are requirements now that adverse events are coded in MEDRA, and with the use of electronic reporting, it's much easier to process this information compared to when they were purely coming in by paper. The next slide shows the form that I mentioned at the bottom, the 3500 form. And this shows you what needs to be filled out by the sponsor. And at the very least, one needs information such as the patient identifier or number, what drug products you're referring to, a description of the event, and the signature of the reporter. But as you can imagine, even with this information, it's very difficult to tell causality of the event. One thing that helps here is that since you have millions of reports, generally those large numbers help, whereas clinical trials may have just thousands of numbers, but the data quality is much higher. When the group, the OSE folks get this information, or even companies get this information, they engage a certain search strategy to see if a safety signal is arising. The first thing they do is decide what specific question they would like to focus on, and then develop a search strategy. And the OSE folks also use the SMQ tool, or they develop what is known as a case definition. So if they're interested in a specific diagnosis, they determine which criteria are important for that. Or they could come up with any number of different custom search strategies. Here's an example in real life of how safety data mining is used for monitoring. And this has come from a publication. This is not just a tool for the regulatory industry, but is one that's used very widely by the regulated industry, and also people in academia use it. Highlighted in this slide is the middle row that has about 8 boxes of different analytical techniques with many acronyms, such as Poisson method for very rare events, or on the right side there are Bayesian-based techniques to analyze safety data, or on the left side very crude frequency-based techniques based on case reports. So all of these analytical techniques are used to see if there are signals, and then if an event is of interest, then extra monitoring is applied for that sort of safety event. I'll close the lecture now with just a few slides emphasizing that information on this topic is available from many guidances from the FDA. I've highlighted 2 of them here that I find very useful. The safety reporting guidance on the bottom is being revised, and I would say within a year, what will be available will be very cutting edge and up to date. This is just a picture saying the same thing. One of the tasks at the agency, besides reviewing this information, everyone is really involved in producing these guidances for industry. And so one should be on the lookout for new guidances, and as I've mentioned, they're updated on a regular basis. So we'll close with this slide emphasizing how important it is to have good quality and standardized data, because as you can see, if you're not on the same page, you will not just be analyzing adverse event, but your analysis itself may turn out to be an adverse event. Thank you. I hope you found this information valuable. If you have any questions, please contact the program coordinator.